Hey, how are you? Can you hear me okay? It's kind of windy where I'm. Okay, perfect. I was scared that it wouldn't work well. How are you all today? Hi, Frank, how are you?
I'm fine. I just put something in the oven.
Yeah,
Yeah.
I was testing if you can hear me well because it's really windy where I am. But apparently it works out fine, so I'm glad. Hi, everyone. And the audience will start in around 4 minutes. So thank you for coming. Uh, really looking forward to this room. It will be really interesting. So thank you, everyone.
It's happy to be here. These style of rooms are amazing. Most often, people have to pay good money to hear from these people and learn from these people. So I'm just grateful and happy to be here and be able to learn.
Yeah, it's so generous of the speakers. I agree to just come here for for free. To just they just come here, take time to make this PowerPoint presentations for us, and to just take the time for no pay. Like there's nothing they'll get out of it really, because
Well, I
most
would just.
speakers that we invite already have a large media attention, and they have already big grants, and um, they really they most of them don't really need this, but they do this just most of the time just because they think it's a good thing that we are doing here, and they want to support this mission of science education for a broad audience and things like that. So yeah, it's really nice that they do this.
Yeah, that point of the education. Uh, that was one of the points that I was thinking about, and still publicity. I think most scientists do work in science because they are working towards a greater truth, and so we as scientists, I think that this audience, you have such a great audience here, and the interactions and discussions. I mean, it's interesting to to go to such a medium like Clubhouse, where you can just look at how do these things work. And I mean, you can literally do this from everywhere. And when I came to Clubhouse, I was thinking this is like a 24-hour conference. And so if you're a scientist, you definitely have something going with publicity and truth to be known around. So there is definitely some huge reward to a great audience.
Yeah, you're right. That's definitely true, Frank. Thank you for saying that. And Olivier, welcome. Olivier is one of our guest speakers. I invited you to speak. So if you accept the invite, or if you click on your profile picture here on the room, you can come up here and speak with us. If you click on the profile picture, there should be an invitation option, accept invite to speak. So if you would do that, we can hear you when you say something. Thank you, Olivier, for coming.
Reads like a French name. I would say Olivier, but probably I'm wrong. I'm interested.
I don't know if you can hear us or if you can read the chat. there I say that I invited you to speak. Let me. So if you when I invite, like right now I'm inviting you, you should get the green bar on the top of your screen where it says accept invitation to speak, something like that, and then you should be able to join us on the stage. Thank you.
The other way to do it, Olivier, would be to tap on your own profile. So if you just see the screen and you see the OG of your profile, you tap on that, and then down at the bottom of that pop-up screen that comes up there you could also tap accept invitation to speak, and then you would also appear on the stage with us.
Hi, Jean-Marie. How are you today?
Hi, I'm good. I don't think you can hear
Yes,
me
yes,
because I
I
don't see the
can.
microphone.
I can
Oh,
hear
you
you.
can.
Yeah.
Okay, good.
When you don't see the microphone, that's when I can hear you. Oh, and
Okay,
your
good.
colleague arrived. Olivier is also here. Hi, Olivier. Nice. I hope I'm saying your name right. French is the major European language. I cannot speak. I'm really sorry about that. I had Latin in school instead of French. My parents' fault. No, I had enough time to by now to learn it, so it's not my parents' fault anymore. So all the way on the bottom right hand, there is a little microphone symbol. So if you press on that, you should be able to unmute yourself, and then we can hear you. If it doesn't, oh, there you
Yeah,
go.
now it should work.
Yes.
Yes.
How are
Welcome to the
you?
stage.
Hi, fine, thank you.
Thank you for coming, and so nice to meet you on Clubhouse. We met
Yeah,
through email. Now we added the voice. Little by little, we get to know each other. Okay. Yeah, thank you so much both for coming. It's such an honor. I posted the presentation on top of the room, and I will right now add the bioarchive paper in the chat so people can access that too. And if we are ready, we can start. If that's good.
Yeah, I think we can start. Let me open the slides on my laptop, and maybe we can give a few seconds for
Yeah, I'll introduce you.
people to.
But I'm sorry, but I don't have like too much information about Olivier. So how about this, Jean-Marie? I introduce you, and if you could say a few words to introduce Olivier,
perfect.
then we start. Is that good? I'm
Yeah,
sorry.
no
Yeah,
problem.
sounds
Perfect.
good.
Okay, welcome everyone to the Science Society, and of course a special welcome to our guest speakers here, Jean-Marie and Olivier. And let me introduce you first, Jean-Marie, and then we'll go from there. He,
Okay.
Jean-Marie, he went to the University de la Réunion and he did his where he studied biology, um, and then he did his master's at La Rochelle University, and he did there studied environment spaces and marine and biology and ocean biology. And then later he also went and did his master's at the University de Antibes, where he studied biodiversity, ecosystems biology. Um, and there he also did his PhD in the physiology and biology of our organism population interaction. And he now uses classic and cutting edge imaging approaches in conjunction with sequencing technologies to investigate unusual life strategies and symbiotic interactions in uncultivated bacteria and small eukaryotes. So thank you so much for coming. I'm very, very honored to having you here. And thanks for introducing your colleague.
Sure. Thanks for the introduction. So Olivier is he's a full professor at the University of Antilles in Guadeloupe, in the Caribbean. He's a marine biology scientist. He's done his PhD at the University of Antilles. I think then he went for a postdoc in San Diego at the Scripps Institute for a little while before coming back for a faculty position at the University of Antilles. And he's now the head of the marine biology lab over there. And he's leading a group. I'm not sure how many people are in your group, Olivier, anymore, but probably 6, 7 at least. Yeah, 10
with a student.
10 faculty assistant professor and some PhD students, and yeah. So and I should say I did my PhD in Olivier's lab actually at the University of Guadeloupe. Yeah. So I don't know. Do you want to add something, Olivier, or I was not really preparing
No, it's perfect.
anything
No problem.
to introduce
Yeah,
you?
it's fine.
Okay, good.
Yeah, thank
All
you.
right.
And usually our tradition is to first ask a couple of interview questions, if that's okay with the two of you. We think kind of it's interesting for a broad audience to learn a little bit of how it is to become a scientist. And our first question would be, how did you choose the path of science? Was it something from childhood on you always wanted to do? Was there some experience that like elicited or sparked that interest, or some teacher, some book? We always think that's very interesting to learn from our guest speakers. Thank you.
Olivier, do you want to answer that first?
Oh, you can go first.
Okay. All right. Well, in my case, to be completely honest, the way I ended up studying biology in the first place is that I was living in Guadeloupe, which is a tropical island in the Caribbean, and when I finished high school, and I had no idea what I wanted to do, and I was very much into diving and snorkeling and looking at the amazing coral reefs and all the small invertebrates and colorful species that you can spot underwater in the Caribbean. And I didn't want to leave Guadeloupe, so I looked it up and I saw that biology was an option. You could study biology and marine biology at the university, the local university. So that's how I picked biology in the first place. And then by studying it and traveling around and diving and getting to know a little bit more the marine environment, that became something I was really, really passionate about. And so I guess I got into it by being curious about the marine environment first. So that's it for me. I don't know, Olivier, if you want to tell a little bit how you ended up becoming a scientist.
Yeah, it's quite similar for me. I was born in Guadeloupe, so everywhere is in contact with marine environment, like for surfing or something like that, snorkeling. So yeah, just after the high school, also in the university, I did first internship in a marine laboratory lab, just to have an experience in a lab. And I love it to play with invertebrates and, and the research like that. So I decided to try to be a scientist too. Yeah. But at the beginning, it's just in contact with marine environment.
Yeah. So in my case, I can add maybe that there was, because you asked that specifically, there was not a plan from the beginning on to become a scientist. It was more something that grew with the years, and it was really one step at a time. Getting into this internship and then this project and discovering these different techniques that are used in the lab to study organisms. When I joined Olivier's lab, first for an internship, And then later for the PhD, I received trainings in microscopy, and that was a big part, a very important step also, because suddenly using light and electron microscope, I was able to really, see what was inside of living organisms, inside of cells, and that was so fascinating for me. So that it became very clear that I wanted to do that more. So that's how it went.
Yeah, thank you for sharing that. That's really interesting. It's so interesting how all the different scientists have like this different paths, but then in the end, it's really driven by curiosity and like passion and curiosity. That like
Absolutely.
yeah.
Yeah.
So it's always wonderful to hear that. I think because then it's such a genuine thing to do, and it's also so lucky, right? We are so lucky to be working in something that we actually are, curious about. So,
Yeah,
yeah.
definitely.
And how did this project came about? Like, is there maybe, like, how did you come and going into this field, or is there a story about this project? Maybe it was really hard to get the grant, or, is there maybe some background story?
Yeah, that's an interesting question. I think Olivier could answer that. I think there was actually originally no grant, and that's one of the reasons why it was a story that built up slowly, right? Olivier, you wanna tell a little bit about how it
Yeah.
started?
Yeah. So I did a PhD on microbes and sulfur-oxidizing symbiosis in between bacteria and marine vertebrates. And after this PhD, when I came back in Guadeloupe after my postdoc in San Diego, I was working on mangrove and seagrass bed, still looking for symbiosis in invertebrate living with bacteria. And after that, I found some biological models with ectosymbionts, and the invertebrate with ectosymbionts with sulfur-oxidizing ectosymbionts are usually white. So I am looking in the mangrove, I am looking for white specimens, like white filaments, some white things. And I found this white long filament attached to leaves of mangrove trees, sunken leaves. So I collect them, I brought them back to the lab, and I decided to look at them just by curiosity first. They are so long, so that I believe it was something like fungi or I don't know, a big, large protists, but not bacteria for me, not bacteria. It was too big to be a bacteria. So I look at them with a TEM light microscope, and I saw no nuclei, no mitochondria, nothing, no organelles usually present in eukaryotic cells. So I said, yeah, this is very strange. Maybe it's a prokaryote, but it's very strange. And it was not my first focus. I was working on symbiosis between bacteria and bivalves. So I just put this on the lab and say, okay, if I will time after, we'll have a look to this. And we work like this, and I spent 5 to 6 years before to work again on this white filament. And after that, we saw that it was a bacteria. And we spent again 5 to 6 years. So it's not a long work on this bacteria. I found them I found the first pictures were taken in 2005. So it's a long story. But we really work hard with Jean-Marie these 2 last years on this model.
Wow, that's interesting to learn. It's so important, I think, to learn. We read the paper, and I think if you're not in the field, then you don't understand how much work there is behind this one paper, and how much trouble and effort many times. So thank you for sharing that story with us. And yeah, now the stage is yours to present this data, and then it's your option if you want to if people can ask questions in between, but most of the time we just wait until you are done with the presentation, but it's really your choice. And thank you, and the stage is yours.
All right. So I think the way we're gonna do is I'm gonna present and I'm gonna refer to some of the slides for those who have the possibility to look at the slides. I wouldn't mind having questions directly during that presentation. I'm also fine with having Q&A at the end, so totally up to you. But because I'm not completely sure of who's listening and what is their background, maybe it's okay to have questions at the same time. So if I'm too technical, or then I can adapt my speech, or on the contrary, if it's too shallow, then I can dive a little bit deeper. So let's try to make it a little bit interactive, if that's okay.
Yes, sure. Okay, thank you.
All right. So how much time do you want me to present? 20, 30 minutes? Is that too much, or do you
No,
want?
it's fine. It's really up to you how long
Okay.
you want to stay. Like, we are
Okay.
here
Okay.
for you.
All right.
Okay, thank you.
So let's start. So I'm going to try to give an overview of the key findings from our study that are in this paper. Actually, on the slide number 1, there's a link to the paper published in Science 2 months ago now. And before I dive into this, I would like to give some introduction, some context. So if you go to the slide number 2, this is a recent representation of the tree of life, and as you can see, all life is in either of these 3 groups, either bacteria, archaea, or eukaryotes, right? And they are color coded, and you can see how diverse are each of the 3 groups. Now, just forget about the tree and maybe picture yourself, going out there in nature and trying to assess the extent of biodiversity around you, just using your naked eye without the aid of a microscope. If you look around, you're going to see, this amazing biodiversity of things like trees, bushes, fungi, birds, mammals, reptiles, a lot of insects, maybe some fish. But all of these fascinating biodiversity, when you look now at the tree of life again, this is all eukaryotic species. All of these macroscopic life forms are just this blue branch, the small branch of the tree that is the eukaryotic group. And this is quite fascinating, right? Like this small branch of the tree of life is actually taking up all the macroscopic space. So now if you move to the slide number 3, another way to look at this is to look at all the different sizes of living things. And if you consider from the smallest ultra small bacteria, which are just a fraction of a micron on the left of that scale, of that linear scale, from the smallest ultra small bacteria to the largest sequoia trees, for instance, that we have here in California, there are 24 orders of magnitudes in terms of biovolumes. That's a trillion times a trillion. That's a very big range. And along that range, as you can see on the slide 3 and 4 now, most of those life forms are complex eukaryotic organisms. For some reason, the bacteria and archaea, they are restricted to microscopic sizes. And this sounds like a simple statement, but it's actually quite puzzling for evolutionary biologists, because eukaryotes, macroscopic eukaryotes, they have emerged, they evolved from archaea and bacteria to some extent through symbiosis. So these complex macroscopic life form evolved from simple microscopic life form only once in about 4 billion years of Earth history, right? And this is very surprising, because until today, all the key ingredients of complex macroscopic life are still present individually in different species of bacteria and archaea, like cell differentiation, endosymbiosis, phagocytosis, compartmentalization, or even multicellularity. All of these individually exist in some bacterial species. But the jump to complex macroscopic life apparently happened only once in the evolution of life. So what underlies the origin of biological complexity is a very big question in biology that has not been answered yet. And another way to put this question is why our bacterial and archaeal cells remain essentially small and simple. So that is kind of the context of our project. So if you move to slide 5, I'm going to now tell you a little bit about what we think limits the size of bacteria and archaea. There are a couple of things that have been proposed and lots of papers about that. So there are 3 things I want to talk about. First, we think that bacteria and archaea are limited by the way they transport molecules. They lack active transport, so they rely completely on something called chemical diffusion. It means that the molecules, all the ingredients of life inside of bacteria and archaeal cells are just moving around randomly and mixing by Brownian motion. And this is very efficient for small distances, along micrometers. That works very fast and very homogeneously. But as soon as you consider a larger cell, like 10 micron, 100 microns, or a millimeter, then this type of transport is extremely inefficient. So we think that this is one of the main reasons why bacteria and archaea have to remain small. Eukaryotes, however, have developed a more elaborate way of transporting and compartmentalizing their biochemical reactions. They can pack biomolecules into vesicles and transport them along microtubules using motor proteins. So that was the first thing. Then the second limitation for bacteria and archaea cell size is something called the energetic cost of genome complexity. So let me explain that a little bit. There's on slide number 6, there's a plot where you can visualize the genomes of viruses, archaea, bacteria, and eukaryotes that are plotted in terms of genome size and number of genes. But even if you cannot look at the slides, the main idea is that simple organisms have small genomes with little genes in them, such as viruses. Then you have some intermediary complexity with bacteria and archaea. They have genomes that are about 3, 4, 5 megabases. They have a couple of thousands of genes. And then more complex organisms, like eukaryotes, have much larger genomes with many more genes. So higher complexity requires more genes and larger genomes to accommodate those genes, right? And there's an energetic cost of having a larger genome and producing many more proteins from more genes, because protein synthesis is actually 75% of the cell's energy budget. So being more complex, a more complex cell supposedly has a much higher energy need. And energy is produced quite universally through the production of a molecule called ATP, which is produced by a small molecular machine that is called ATP synthase. And ATP synthase is restricted to membranes. It's not floating freely inside of the cell. It has to be attached to a membrane. And for bacteria and archaea, it is proposed that energy production by ATP synthase is mostly restricted to the cell membrane. So the surface area around the cell is the surface that produces the energy of the cell. And again, that is fine for small cells. If you consider on slide 7, if you look at slide 7, if you consider a small 2 micron bacteria, the surface area of membrane is large enough to sustain the energy need of the cell. But if you consider a much larger bacteria, let's say 100 micron bacteria, the surface area to volume ratio drops about 50 times. And then suddenly there's not enough membrane. At least at some point, if you consider larger bacteria, there's not enough membrane surface area available to produce all the energy that the cell is going to require. So that is the second limitation, a theoretical limitation that is proposed for the limit on bacterial cell size. I'm going to skip slide 8 and just go to the third limitation, which is something that was proposed in 2016 by Kempes and collaborators. It's called the ribosome catastrophe. It's a very interesting model that was published by this group. They looked at all kinds of model systems of various sizes, and what they did is they built a model where they try to predict how much space is going to be taken up by the various components, the ribosome, the DNA, the membrane, the proteins, everything that's in a cell. How is this going to evolve when you consider either smaller or larger cells? And I'm not going to go too much into detail, but what they came up with is a predicted maximum cell size above which the number of ribosomes that are needed for the cell to function is more than the available space that there is in the cell. So they call that the ribosome catastrophe, and that for them sets the upper limit of bacterial cell size. So there are some exceptions on slide 10. I'm not going to spend time describing them, but there are some giant bacteria. There are exceptionally large bacteria in at least 4 different groups. One of them is Thiomargarita. There are already known giant bacteria in the Thiomargarita group, which is the same group where our giant Thiomargarita magnifica belongs. And if you go to the slide 11, that will give you some kind of overview of everything I've said. There is a theoretical framework that predicts a possible range of cell size for bacteria, with a lower bound and an upper bound. And indeed, most bacteria that are studied in the lab, they are falling within that range. But there are some exceptions that are challenging the upper bound of bacterial cell size, And spoiler alert, the giant Thiomargarita magnifica that Olivier discovered in mangrove is actually 2 to 3 orders of magnitude above the predicted upper bound of bacterial cell size. So our hypothesis was that these giant Thiomargarita magnifica bacteria, they may have a higher biological complexity that is not taken into account in those models, which allow it to break free from some of the biophysical and bioenergetics limitation on cell size. So that was a kind of the context, right? I'm gonna now go briefly over four bullet points that we develop in the paper. I'm gonna first tell you why we think that these giant long filaments are single cells. Then we're gonna talk about the fact that they are more complex because they have their DNA contained in a new type of membrane-bound organelles. We'll talk a little bit about the genome, and then finally about the way these giant cells produce energy. So on slide 13 and 14, you have 2 really nice pictures provided by a colleague in Guadeloupe, Pierre-Yves Pascal. This is the sampling site. So on these pictures, you can recognize the mangle tree, and at the top of the sediment, all the decaying organic matters that is covered with white sulfur bacteria that are feeding on the hydrogen sulfide that is diffusing from the sediment. So this is the environment where the Thiomargarita magnifica cells grow. They are marine filamentous bacteria. We cannot see them on those pictures, but it gives you an idea of where they grow. On slide 15, however, you can see how those giant bacteria look like. So they are white filaments that are developing on a dead leaf. That's what you can see on the right. They can grow up to 2 centimeters, but the average size is about 1 centimeter, 9.72 millimeters to be precise. That's what you can see here on the left. They are about the size and the shape of an eyelash. And the first observations that were done immediately revealed that they were apparently not segmented. So they were apparently composed of a single cell. So a big part of the paper is about producing or showing convincing evidence that these filaments are indeed just giant single cells. So if you go on slide 16, sorry, there's a link to a YouTube video. I don't know if you can.
I would also have a question here, Jean-Marie.
Sure, go ahead.
And that is, are there any? I'm not. Biology is very far from me, so this is a very naive question. But are there any differences whether bacteria or what have you are growing in air or in water? Is there something to the medium that is kind of relating to the possibilities of the sizes that you talked about? That's something that I just came up as a question for me.
Yeah, thanks. That's a good question. It's a good question. Actually, there is something specific about the environment these bacteria live in. Indeed, so they live. If you look again at slide 14, they live at the interface between an anoxic sulfidic sediment and oxic water. So they position themselves exactly at this interface where there's a very steep chemical gradient between no oxygen and very high hydrogen sulfide from the sediment, and no hydrogen sulfide and high oxygen from up above in the seawater, and that makes it a very specific environment where there's a lot of free energy that's available to them, because if you can harness the redox potential between these reduced hydrogen sulfide and the oxygen from above, there's a lot of energy that's available for their metabolism. And free living bacteria, if you were referring to small microscopic bacteria that live freely in the water, they have this challenge that if they want to exploit this gradient, this chemical gradient, they need to be exactly at the right interface. But they are washed away by water currents or waves or whatever you name it, while these giant bacteria maybe, I mean, this is speculation here, we have not studied that, we don't have data to support this, but there are papers that are saying that maybe giant bacteria in general, large sulfur bacteria in general, have this advantage that they can exploit better the chemical gradient between the sediment and the seawater. So that could be one thing. Does that answer your question?
Well, yeah. Thank you so much.
Okay,
continue.
good. All right. So going back to slide 16, if some of you clicked on the link for the YouTube video, you can see a 3D animation of a 3D rendering of a single filament. So what we did basically is Olivier collected samples in mangrove. He fixed them with chemical fixation, stained the membrane with some specific fixatives, and then he shipped them to me in California. And what I did is I basically did a CT scan of these bacteria. We placed them in an X-ray microscope, and we looked at them with X-rays, pretty much like medical X-rays, but a very specific type of X-ray microscope. And then we reconstructed the 3D object from these X-ray two-dimensional projections, and we ended up seeing, like you can see on the video, that the filament is indeed a continuous tube that doesn't have septation that would make it a multicellular filament. So that was the first technique that really pointed to the entire filament being a single cell. Then we used a second technique. That's what you have on slide 17. There's also a link to another video if you want to click, but I can also explain if you're not seeing the slides or the video. It's basically we used a membrane dye to stain the membranes, and we observed entire cells with confocal laser scanning microscopy. And we reconstructed these entire filament in 3D, and again, we observed that the entire cell is made of a single continuous cytoplasm with a single membrane that is continuous all along the filament. And finally, we used transmission electron microscopy, which Olivier had done extensively already before I started to do, but we observed long longitudinal sections. And with electron microscopy, you have the possibility to zoom in much more into the ultrastructure of the cell. And again, we could not see any membrane that would separate the filament into several cells. So we confirmed with 3 different techniques that these filaments were indeed ginormous single bacterial cells. So that's for the first point. And if you go to slide 19, there's one of the figure that's in the paper on the left that shows you on a log scale where Thiomargarita magnifica is in terms of in length. So here it's a length of cells or organisms and not biovolume. But you can see that it is much bigger than most bacteria. It's in the range of centimeters and not micrometers. It's actually larger than some complex multicellular organisms such as Rabitis elegans worms or Drosophila, which are famous model systems, eukaryotic model systems. So that's for the first point. Then we looked into the inside of the cell, and we used again a variety of microscopic investigations to try to understand what makes it possible for these cells to be so big and to sustain such a large complex body structure. So we stain the membrane on cross sections of cells together with DNA, and that's what you have on slide 20. So you have a cross section of a cell, and we did label the outer membrane around the cell in orange using a membrane dye called FM143X. But we also were surprised to see that there were also membranes, structures that have membranes inside of the cell, in the cytoplasm, surrounding the large central vacuole. And inside of these membrane-bound structures were also labeled DNA. So that was a very big surprise because apparently in these bacteria, the DNA was contained in a membrane-bound structure that we can call an organelle, which is something that is not typical for bacteria. It's actually a characteristic of complex eukaryotic cells. But in bacteria and archaea, the DNA is known to be always free-floating in the cytoplasm, if you will. So, sorry, I don't know if I got disconnected or what.
No, you're
hear me?
completely fine. Yeah.
Okay, good.
Absolutely.
My phone did something funny. So then if you go to slide 21, we looked at where are the ribosomes of the cells, and we used a technique called fluorescence in situ hybridization, which consists essentially to apply a fluorescent dye to the ribosomes. And as you can see on the image, the ribosomes of the cells were labeled with this fluorescent dye, and they colocalized with the DNA in the pepins. So in the organelles that contain the DNA. These organelles that, by analogy with the small seeds in fruits such as kiwi or watermelon, we named them pepins after the French word pépin. And these pepin organelles, they were present throughout the entire cell, and they were containing not only the DNA but also the ribosomes of the cell. And we confirmed with transmission electron microscopy that they were also surrounded by a membrane. So that's for the second main point of the paper, that not only are these cells giants, they are also displaying a higher level of complexity in the sense that they have compartmentalized their DNA and their ribosomes into a new type of organelles that we named pepins. And then at this point, we asked a question. We asked ourselves, yes, okay, so these giant cells are centimeter long. That's crazy. That's unheard of. But maybe these gigantic cells are not really alive throughout the entire length of these filaments. Maybe it's just some kind of dead or inactive stalk that is supporting the active growing tip of the filament, and that would make a completely different story, right? So without going too much into detail, we used a technique called BONCAT, where you incubate the live cells into seawater, and you provide them with a molecule which will be taken up by the cell and integrated into newly produced proteins. And this molecule, once it's integrated into protein, you can label with click chemistry. You can label it in fluorescent color. Here on slide 23, it's been labeled in green. And what we saw at the end of this BONCAT experiment is that the entire length of the centimeter-long bacteria was active in terms of protein synthesis. So we were not in that scenario that we had a long inactive cell supporting a growing tip. We were really looking at a actively synthesizing giant microbe. That brings me to the next point, which is the polyploidy, the genomic investigation. So if you go to slide 24, we started to to investigate how is structured the genome of these cells, because in a regular bacteria, there's usually one or maybe sometimes a few copies of the genome that is free floating in the DNA, and that is sustaining the entire cell. But here, because of the size, of course, you cannot imagine that a single genome copy could be sustaining the entire giant cell, right? So like actually all large bacteria, Thiomargarita magnifica is polyploid. It contains many copies of the genomes that are, in this case, contained in pepins. We estimated the number of copies to be, for a fully 2 centimeter long cell, it could be up to 738,000 genome copies, which is a very big number for a single bacterial cell. And then we sequenced that genome. We managed at the Joint Genome Institute, which is a sequencing facility of the Lawrence Berkeley National Lab, where I work. We sequenced 5 draft genomes from 5 single cells, and what we found out is that these giant cells have a very interesting genome that is quite large. It is about 3 times larger than most bacterial genomes. It also contains many more genes, about 11,000 genes, which is again 3 to 4 times higher than most bacteria. What's even more interesting is that we found out that there's about 25% of the genome of these cells that is dedicated to secondary metabolism. That's on slide 25, this BGC colon. BGC stands for biosynthetic gene clusters. Those are the genes that are indicative of secondary metabolism, such as producing antimicrobial molecules or molecules that are involved in complex pathways, such as communication, complex behavior, social behavior, that kind of stuff. So this was a very interesting finding as well. Then if you move to slide 26, I think that's going to be my last point about the genome. We also found out by looking and analyzing the genome of Thiomargarita magnifica that it lacks some of the essential genes for cell division. and in contrast, it was having a full complement of genes for cell elongation, with some of those cell elongation genes that were actually present in multiple copies. Some of the elongation genes were duplicated. So on one hand, we have a giant cell that does not divide like other bacteria through binary fission, like most bacteria in archaea. Instead, it grows into a long, centimeter-long filaments, and indeed, it is missing some of the essential genes for classic binary fission cell division, and it has an extended complement of genes for cell elongation. So that was a very nice bridge between the genome analysis and the cell morphology analysis. Okay, and now I'm gonna switch to the last point, which is how energy is produced in these giant cells. And to go back a little bit, if you remember in the introduction, I told you how bacterial cells may be limited by the way they produce energy. If you look now on slide 27, there's a schematic representation of a small cell that is 1 micron radius. If you think of a larger cell that is a 3 micron radius, the radius increases 3 times, but the surface area of membrane that produces ATP increases 9 times. And at the same time, the volume of the cell, which is essentially a proxy of the energy need of the cell, increases 27 times, just because of the surface area to volume ratio, right? So you can see that increasingly large cells have an issue that they are not increasing their surface area as fast as they are increasing their energy demand, right? So the question for these extremely big Thiomargarita magnifica cells, the question was, yeah, but how do they produce their ATP if ATP production is restricted to the cell membrane on the surface of the cell? How do they produce enough ATP to sustain such a large biovolume? So we label the ATP synthase to localize bioenergetic membrane, to localize ATP production on cross sections of cells. And instead of labeling the surface of the bacteria, we labeled the entire complex network of membrane that was present intracellularly inside of the cytoplasm, including around the pepin organelles. So that was a very interesting observation that supports somehow the idea that to grow to larger sizes, you need an expanded network of bioenergetic membranes. And I think that's the last main point I wanted to tell you about for the paper. There's a summary slide on slide 28, and very briefly to sum up and conclude. So we show in this manuscript that Thiomargarita magnifica are by far the largest bacterial cells ever observed, that they have a higher level of compartmentalization because their DNA and ribosomes are compartmentalized into structures, organelles that are membrane bound, and we name them pepins. They have a large genome with more genes than average bacteria. They are missing some essential genes for cell division, and they have duplicated genes for cell elongations, and they have this expanded network of ATP producing bioenergetic membranes. So altogether, these giant cells show a gain of complexity and somehow challenge the traditional concept of a bacterial cell. And we think that investigating further the biology of Thiomargarita magnifica, and in particular its energy metabolism and the precise role of the pepins, may be helping us in the future to better understand the origin and the evolution of biological complexity. And with this, I'm done. So there's I guess some time for question if there are questions, and we'll be happy to answer them.
Thank you so much, Jean-Marie. This was an amazing presentation, and it's so interesting. The question I have is, like, I have various questions, but I don't want to, I will give the next question to somebody else. So does this change basically the origins of, of our cells that we have with the nucleus? Because, a lot of times I heard about the archaea, and we had the guest speaker here previously, I think last week or the week before, I think 2 weeks ago, talking about his work with archaea from like the deep ocean, and he works on these Asgard viruses, and the theory is that they kind of helped develop this nucleus that we have. Like, do those bacteria that you found, do they also have these type of viruses in their environment, or do you think that they could have developed, do they have developed independently without the help of viruses or so on, like nucleus and things like this?
All right. So there are basically two questions in your question. The first that relates to, do they have viruses that may have somehow helped them evolve some structures like the pepins? Or we don't know anything about viruses that would be infecting those bacteria. We have not looked at that. We don't know anything about viruses that would be associated with these bacteria, so that I don't know. However, the other part of the question, which is, does this finding changes? Does it change anything in terms of the origin of the eukaryotic cell and the origin of the nucleus? And the answer, thanks for asking this question. That's a very important point to clarify. So the answer is very clearly no, it doesn't change anything, because what we, so if you go back to the tree of life, if you go back to the slide number 2, and you see the eukaryotes that are indeed branching into the archaea domain. They have evolved from Asgard archaea, apparently. The origin of eukaryotes is completely distinct from Thiomargarita magnifica. Thiomargarita is a bacterium. It's from the group in yellow here at the top of the tree. It's a gamma protobacteria, so they are completely unrelated to eukaryotes. If you want to think about how come that they have these structures that contain the DNA in a membrane-bound structures, that somehow functionally it's tempting to think about that as an analogy to the nucleus in eukaryotes, but it's really a different type of structures. It also contains the ribosomes, while the nucleus compartmentalize the transcription from the traduction that happens in the cytoplasm of the eukaryotic cell. So here, both transcription and traductions happens within the peptide. So there are very different structures. But the fact that the DNA is protected in a structure with a membrane is, I'll give you that, is somehow analogous to the nucleus. But if anything, this is convergent evolution, and not it's not related to the Asgard archaea that evolved into the eukaryotic cell.
Thank you. Anthony had the questions from early on from the talk, so welcome to the stage, Anthony, and please ask your question. Can you hear us, Anthony? To unmute, it's all the way on the bottom right hand. There's a little microphone symbol, so if you press that, you can speak with us.
I have a quick question if he's not able to.
Yeah.
This is kind of a joke, but do we need to change the name of microbiology departments?
Yeah. Well, that's an interesting thing, right? Like it's when I'm asked to describe myself, when I've been talking to press or stuff after the publication, they ask me if I'm a microbiologist, if we're still microbiologists when what we are studying is actually not microscopic. So yeah, we might have to come up with a new names for giant microbiologist. Yeah.
Yeah, and I don't know whether maybe this is. Do you think this could be the tip of the iceberg? Maybe we just haven't been looking for them enough.
So maybe not the tip of the iceberg. That would I wouldn't go that far, but definitely there is this idea now that I mean, giant microbe were known before, right? I have not acknowledged that enough in my speech before, but there were some giant bacteria that were known. Some of them were visible to the naked eye already. They were about 50 times smaller than this one. So we are pushing again the boundary a little bit further. But the question whether or not there are other giant microbes that are awaiting discovery, yeah, I think probably, because the way microbiologists sample bacteria is just not adapted to finding these giant microbes. Like most marine biology, microbial ecology sciences, they would go and collect water and filter, exclude large things because they don't want the eukaryotic contaminants in their samples, and then they would filter microscopic cells on a 0.2 micrometer filter, and they would sequence the DNA. So if you sample bacteria like this, you will miss all the giant microbes. So now, how many giant microbes are out there that we have not recognized? That we don't know. But I hope that this paper will encourage other groups to look for other giant microbes. That would be really interesting to see other species found in various environments.
Yeah, that's really cool. You don't find what you don't look for.
Exactly.
I think Anthony is ready now for his question. Go ahead. Okay, maybe not. Maybe I misunderstood in the chat. Dr. Shah, Einer, please, Frank, Gilbert,
Yes.
please ask your question.
Yeah, thank you so much for sharing your research with us. My question is there any phosphate accumulation that you found? Also, is there any differences between the pH of the vacuole and, cytosol? You can answer this one and I will go forward for the next one.
I didn't quite understand the question. I'm just going to repeat to make sure. Did you ask, is there any difference in terms of pH between the content of the central vacuole and the
Yes,
cytosol?
that was one of them. And another one was the phosphate accumulation. Is there any phosphate accumulation that you found?
Okay, so phosphate accumulation, I don't know. We have not investigated the phosphate metabolism. I know that I think there are some papers about phosphate granules in other Thiomargarita species. We have not looked at that, and the very obvious granules that were present in the cytosol of Thiomargarita magnifica were sulfur granules. So that has been characterized by Olivier's lab in Guadeloupe. They used electron microscopy coupled with elemental analysis to show that these large spherical granules in the cytoplasm were elemental sulfur granules. I don't know about phosphate accumulation. We have not looked at that.
Jean-Marie, yeah, we got no signal for phosphate.
Okay,
Phosphorus.
so there might not be phosphate granules. Yeah, we don't have evidence that they are phosphate granules. I guess we can put it this way. And then concerning the pH of the vacuole, we have not measured the pH with like microsensors, or there are some dyes also that allows you to do that. And there's a preprint by another group studying Thiomargarita species. I think they do such techniques, and they have very interesting images, beautiful images about this. We have not done that kind of work. We may have some. I think Olivier has some preliminary data that the vacuole contains nitrate, which is what has been described for most large sulfur bacteria with central vacuole. Not all of them, though. But so we might be in the same scenario where the vacuole contains nitrate, but this has not been published yet. And
No, because it's
and in
only
terms of pH, go ahead. Sorry.
Yeah, it's just preliminary results about the nitrate. But I have some contact with a French lab, and he's also doing using dye to measure the concentration of nitrate or phosphate inside the big cell like that. So maybe in a few months we will be able to say if we really have nitrate and how much, what is the concentration of nitrate inside the cells, and also about phosphate. I hope so.
So
Anyone hear me?
is there any differences between the usage of the protein, I mean, for this species, and for example, in comparison with the E. coli, if you want to explain, because we have amino acid starvation, and I was just wondering about this species and how the species use the protein as an intracellular amino acid pool.
I'm not sure I understand your question. Are you asking? Yeah, no, I don't understand what you're asking exactly. What do you mean by usage of protein?
You mentioned
You mean
about the
what?
protein, right?
I mean, we have. Sorry, what?
You mentioned about the protein that you just
Yes.
found it in this species. I was just wondering the process of the protein when we're just talking about the protein. They're supposed to be in some of the species similar to this species that you find there is differences between how they are using the protein as an intercellular amino acid pool, and we have amino acid starvation that is happened. So, for example, include the tRNA, it become discharge and there is no charge, and after that they are just switching the component that they are using. I was just wondering maybe you just go forward to those details or not.
I don't think we can answer that your question. We have not looked at that. So in terms of protein synthesis, the only thing we've done is to visualize where, to localize protein synthesis inside of the cell with the Boncat approach. We have not analyzed the proteome, so we don't know the nature of the protein that are being produced by these species. And I don't think we can answer your question with the data we have right now.
Hello, this is Anthony after many tries. Finally got my sound working. Yeah, thanks for pointing at the trade-offs and hypotheses about that earlier. That's really interesting. And I was wondering, slightly technically, I was wondering if you think what you think of the idea that you've got these little regions with nucleic acids and ribosomes, and you have this other huge volume that doesn't. I was wondering if what you think about the possibility that could this big space with no ribosomes, no nucleic acids, could it be sort of an enormous shared periplasm, the place between the two membranes that ordinary bacteria have? Could it be sort of like separate cytoplasms in a shared periplasm?
That is a very good question, and we again, we are not going to be able to. Sorry to disappoint. I'm not going to be able to answer that. We have not settled that. It's you're basically asking about the membrane topology in these giant cells, right? And it is very different from model bacteria, that is for sure. And what I can tell you is that we are currently conducting some additional microscopic investigation. We're going to try to use cryo-electron microscopy. We're going to try to look at also other large sulfur bacteria to understand better the membrane topology. We don't know the answer to your question at the moment. There are some structures within the cytoplasm that are also round shaped, that resembles the peptide, but they don't have DNA or ribosomes. So yeah, we also have this limitation that so far all the microscopic investigation we conducted was done on fixed material. We don't have these cells growing in the lab, so we relied 100% on fixed cells for all the microscopy we did. So that means that we always only have a snapshot, in time of how the cell is organized and how it is, right? We don't know how dynamic this is. We don't know how the pepins evolve. We don't know how they are formed. We don't know if they mature into something else. So all of these are open questions at the moment, which makes it very exciting. There's a lot more to understand and to discover about these fascinating giants.
Love it. Good luck.
Thanks.
Yeah, the hour is basically up, but if anyone has some last questions, please post them in the chat or, unmute yourself, like flash your microphone. Yeah, Einar, hi.
Yeah, hi, Katerina. Hi, Jean-Marie.
Hi.
Beautiful. So I'm not schooled in this subject, but I had two questions, Dr. Shah, actually, and your conversation with Dr. Shah gave me answers to one of them. But when it comes to the energy-driven part of this bacteria, most likely driven by light. So this means that there is
Driven by what? Sorry, what did you say?
some form of light, which means that they're driven by some form of symbiotic relationship with the environment, basically attached to atoms and electrons emitting lights when they change positions and go in and out of the nucleus and whatnot. Do you have any thoughts around this? Thank you.
All right. I'll try to understand what you're asking. I'm not sure I understand. So first, what I can tell you is that I have not made that very clear, maybe, but these cells they are not photosynthetic. They are chemosynthetic organisms. They have we have various evidence that their metabolism is chemosynthesis. They are capable of using the chemical energy that is contained in reduced sulfur species to fuel their metabolism and fix inorganic carbon. So they can fix carbon using the energy of hydrogen sulfides. So whether or not they are driven by light, if they have some kind of if they are growing towards light, to just like plants would do to position themselves or grow in the right direction, we don't know. We don't have any evidence that they can sense light. We have not looked at that either. But yeah, but they are not. What we know for sure is that they are chemosynthetic organisms, not photosynthetic organisms.
Thank you. Thank you very much.
You're welcome.
Yeah, let me check again in the chat if there was another question. Someone asked if the genomic data is available in a database for people to check out.
Yeah, absolutely. Everything is available. So if you because you shared the link to the preprint, but it's not in the preprint. However, in the final paper, if you check the final paper, you will find all the accession numbers. So the raw reads have been deposited to SRA, NCBI, and we also made available the pre-binning libraries assembled genomes and also the Thiomargarita metagenome assembled genomes, because we used MDA. So I mean, anyway, long story short, yes, all the data are on IMG and on SRA. So the accession numbers are in the supplementary table in the supplementary material of the paper.
Great, thank you so much. Oh, Warrior, you joined the stage. Is it okay to ask one more question, Jean-Marie?
Yeah, sure. Yeah, thank you.
Okay.
For me, it's I will have maybe 3 more minutes, then I'm gonna have to go. but I have time for one more question.
Okay,
Yes.
thank you.
Yeah, you said that we don't know much about this cell, and probably the pictures you have now or whatever you are, I mean, observing is just like dead cells on a microscope. Yeah, and I think we have lots of theories on how the cell work, including the ribosome and making, proteins from RNA and all that is still a theory. Is it possible that all that we know about the cell is wrong, and we have to start over because
I don't think. No, I don't think that all we know about cells is wrong. Not at all. I don't think. Well, also, I am absolutely sure that we are not just looking at just dead cells under a microscope. I mean, they're dead once we've killed them, but when we collected them, they are alive, and we know that for sure because we have. Olivier's group is working with the live cells. They see them producing these terminal buds. They see the terminal buds being released. We have also the Bancat experiments that are performed on live cells, where we can visualize active protein synthesis in the cells in the course of a few hours or overnight incubation. So there's no doubt that we are studying live, healthy cells, and I don't think we are.
just for example, when we talk about ribosomes, yeah, do we have some facts that really we get mRNA coming from the nucleus and then goes out to the ribosome to create a protein? Is that being proved, or just a theory?
You're asking in general in biology, like that's a general question you're
Not
asking
in
me.
general. I'm talking about right now. They are telling us that the ribosome creates, proteins from RNA, which is copied from the DNA in the nucleus. Did you see
Yes.
that yourself?
So, indirectly, yes. So with the difference that we don't have a nucleus here, we have a structure that we call pepins. That is not a nucleus. We have the DNA inside of the pepins that we can directly visualize with DNA dye. We have the ribosomes inside of that same structure, which we can also directly see with two different techniques. We can label them with fish. We can also recognize the ribosomes under the transmission electron microscope. So we know that
but between them, you said we have the nucleus and we have the ribosome. But
and
hold on,
I'm sorry, but our
hold on.
guest speaker has to go,
Let me finish.
and you're asking questions that are very, I don't know, that are kind of solved questions. I'm sorry, but we don't have too much time, and also if you need some updates on basic biology, we can give you book recommendations and review recommendations. Reach out to me, and I can recommend you literature to read. Thank you so much, Jean-Marie and Olivier, for giving this amazing presentation and for this really fantastic work that you shared with us. I hope you get all the funding to to research this bacteria more.
Thank you.
Thank you so much.
It's so interesting. It's so interesting to me, and I think it's interesting in general. So we wish you all the luck, and maybe one day you come back and give us some updates about this project or other projects you're doing. Thank you.
Sure, it will be our pleasure. Thank you so much for inviting us, and for the very nice exchange and opportunity. Thanks. Bye.
Thank you.
Thank you so much.
Thanks. Bye bye.
And thank you everyone for coming and asking questions, being here, being interested in research and science. And if you like questions like discussions like these, follow the club. We will have more events like this. Our next event is tomorrow with Dr. Liu, who will be talking about new genetic research on who are actually the world's earliest seafarers. I don't know if you know, I'm Portuguese. I'm really curious about that. It's for sure not Portuguese, so it won't be Portuguese.
Probably Chinese. No, I'm just joking.
Or Easter Island, maybe? I don't know. We'll learn from her. We'll see. And then on Thursday, we will have Dr. Gu at 2 p.m. EST, artificial microtubules to transport cargoes. He will give us really interesting presentation about this new type of technology, which I think will be very useful in modern medicine and technology. And then on Friday, we'll have Dr. Pshishya talking about new quantum research results and putting that into context with quantum consciousness theories like Penrose and how he's basically collapsing with his lab results, those theories. Sorry, people, but please come and discuss with us. And I'm looking forward to the rest of the week with everyone. I hope to hear you all back soon. Okay, I'll close the room in 3, 2, 1. Bye, everyone. Thank you.